So my name is Agnieszka Jakubowska, also known as Aggie, and I am a specialist registrar in chemical pathology and metabolic medicine in the east of England. During this session, I will try to cover the basic concepts in mass spectrometry and some commonly used terminology. I am definitely not an expert in mass spectrometry, so the aim of this session is to serve as an introduction into mass spectrometry for new medical doctors entering chemical pathology. So before we start the session, I just really wanted to recommend this textbook for anyone starting their journey into mass spectrometry. The reason why I would highly recommend it is that it makes a lot of quite complex ideas very simple, and it's about 250 pages long. I have also used quite a few diagrams in today's session from this textbook. It's called Mass Spectrometry for the Novice. So first of all, what is mass spectrometry? So mass spectrometers are analytical instruments that can be used to analyze both quantitatively and qualitatively a range of compounds from simple gases to complex biopolymers. They produce a beam of gas phase ions from samples known as analytes, which they then sort the resulting mixture of ions according to their mass to charge ratios. They do so by using electrical or magnetic fields, and they then provide analog or digital output signals, which are known as peaks. From those peaks, the mass to charge ratio and the intensity, which is also known as abundance, of each detected ionic species can be determined. So there are some important principles to remember in mass spectrometry. Firstly, mass spectrometers can only analyze ions, which are electrically charged molecules, and they must be in the gaseous phase. Secondly, mass spectrometers do not measure mass, but they do measure mass to charge ratios. And then Z, which is charge, is often but not always equal to 1. The mass to charge ratio is derived from the balance between the magnetic force acting on the charged particle and the centripetal force required to keep it in circular motion. The relationship is crucial in mass spectrometry, where it is used to identify the mass of ions based on the radius of curvature in a magnetic field. And here you can see the basic sequence of events in mass spectrometer. On the next slide, I'm just going to briefly go through history of mass spectrometry. So the origins of mass spectrometry go back to characterization of positive rays by J.J. Thomson in the early 1900s. Over the next 60 years, there were significant advances in both instrumentation and applications. Applications included the demonstration by Aston that many elements had different isotopic forms. So this ability to differentiate isotopes led to the collection of uranium-235 isotope and its use in the atom bomb. In the late 1960s, mass spectrometry was coupled to gas chromatography, which allowed the separation of complex mixtures so that individual components could be entered into a mass spectrometer. However, there was still a problem with polar compounds decomposing when heated prior to vaporization. And this included a lot of biological compounds. This polarity barrier was partially breached by the development of fast atom bombardment in the 1970s. So this is also known as FAB. Then mass spectrometry was revolutionized in the 1990s when electron spray ionization, also known as ESI, and matrix-assisted laser desorption ionization, also known as MALDI, were developed. I will discuss both of those techniques in more detail later, but they involve release of charged ions in a gaseous phase without heating. ESSI has allowed coupling of mass spectrometry with liquid chromatography, and therefore the analysis of polar bioactive compounds such as drugs and metabolites, which we use so frequently now in the clinical biochemistry lab. And of course, this has led to multiple Nobel prizes being won due to those discoveries. So on this next slide, I just thought I would briefly go through to how mass to charge ratio is actually derived. So when a charged particle moves through a magnetic field, it experiences something called Lorentz force that is perpendicular to both its velocity, which is v, and the magnetic field, which is capital B. The force on the charged particle with charge, which is q, moving with velocity v, in a magnetic field which is perpendicular to the velocity, is shown in the first equation. In mass spectrometer, the charged particle is often forced to move in a circular path due to the Lorentz force acting as a centripetal force. The centripetal force required to keep a particle of mass, which is denoted as m in the equation, in a circular orbit of a radius r, is given by the second equation. The third equation is when we set the two expressions for force equal, and then the equation underneath it is after cancelling v from both sides. So we are simplifying the equation. We can then rearrange for m over q. And in mass spectrometry, the charge q is often expressed as z to represent the charge number of an ion rather than the absolute charge of a single particle, such as an electron or a proton. So the notation Z is commonly used to indicate the number of elementary charges on an ion. In a magnetic sector mass spectrometer, the deflection of an ion in a magnetic field is directly related to the mass to charge ratio. This principle allows ions of different mass to charge ratios to follow distinct trajectories and strike specific locations on a detector. This does not apply to time of flight mass spectrometry analyzers, which I will discuss in more detail on a later slide. And here is a diagram of a mass spectrometer. So mass spectrometers have 6 major components to carry out ionization, mass separation, and detection. And as you can see on the diagram, you first need to have a sample introduction system, followed by an ion source where analytes are vaporized and ions are produced. Then you have a mass analyzers where ions are separated according to their mass to charge ratios. Following this, you need an ion detector where the signal intensities, known as peaks, which I have mentioned earlier, of each separated mass to charge values are determined. We also have a fifth component known as a vacuum system, and you need this to prevent collisions with neutral gas molecules, as well as with the walls of the analyzers and the detector. So the final component of a mass spectrometer is a computer, and it's used to control the conditions of the experiment, then record and process the data acquired. Now I'll move on to some common definitions. So just to start off with, this is a quick recap of the terminology commonly used in mass spectrometry. So as a reminder, ions are atoms, molecules, or fragments of molecules that carry one or more positive or negative electrical charges. Hard ionization methods, such as electron ionization, pass on more energy than that required for the formation of molecular ions. This excess energy causes the dissociation of bonds within the analyte, leading to extensive fragmentation. In soft ionization methods, such as electron spray ionization, the energy associated with added charge is usually only sufficient to generate adapted ions, but not to cause fragmentation. Now, a mass spectrum is a two-dimensional representation of the distribution and abundance of the ions introduced into mass analyzer, which are separated according to their mass to charge ratio and then recorded. The image on the slide shows the basic features of the mass spectra. Energy is added to the molecules during ionization. The distribution of the energy may result in the breaking of chemical bonds and therefore in fragment ion formation, which you can see on this diagram. Sometimes the fragmentation might be so extensive that no molecular ion is observed. The molecular weight of the analyte is often represented by the peak with the highest mass to charge value. So the one furthest to the right, known as a molecular ion. The base peak represents the most stable ion resulting from the ionization process. Therefore, this is the most abundant peak, as you can see in relation to the relative intensity on this diagram. Fragment ions might appear at various masses, but their masses are always lower than that of the molecular mass. As I have already mentioned, the energy added to the molecules during ionization might lead to the decomposition of the molecular ion, as well as fragmentation of any formed ions. Isotope peaks are observed if the analyte includes elements with isotopic forms. The most commonly observed isotope peaks are due to the carbon-13 isotope of carbon. Of course, as we will remember for our A-levels, carbon normally has a molecular mass of 12. So now a little bit about sample introduction. So mass spectrometers operate at high vacuum, so can only analyze samples that are in the vapor state. The neutral analytes must also be converted to ions. The aim of the sample introduction system is to produce vapors from samples and to introduce an adequate quantity of sample into the ion source so that its composition still represents your original sample. Sometimes the sample introduction and ionization process occurs at the same time. And this happens in certain techniques, such as atmospheric pressure ionization and electrospray ionization. I will discuss those in further detail on upcoming slides. There are a variety of methods for sample introduction. One example is temperature-controlled solids introduction probe, which is also known as direct insertion probe. The disadvantage of this sample introduction method is that it has to be inserted through a vacuum lock and a heated volume from which evaporated samples enter the ion source through a control valve. Successful vaporization depends on the polarity and molecular mass of the analyte. So for small molecules, less than 1 kilodalton, polarity is the most important factor, particularly the number of primary amino acids, hydroxyl and acidic groups that are present. When there are multiple of those groups present, the compounds will often be destroyed by heat before they are vaporized. Now, chemical derivatization is the conversion of an alcohol to its trimethylsilyl ether, or the acetylation of an acid group, which can be used to reduce the polarity and increase the volatility of analytes, which increases the chances of obtaining useful spectra. So that can be another technique applied for sample introduction. When the analyte is larger than 1 kilodalton, heating is likely to decompose the molecule. So this is why we have combined sample introduction and ionization methods in both MOLDI and API, which I will discuss further in upcoming slides, and that resolves the problem of sample decomposition. So samples of biological origin mostly require chromatographic separation. with subsequent sequential introduction of the species into the ion source. Gas chromatography, liquid chromatography, and capillary electrophoresis can be interfaced directly with mass spectrometers. And the purpose of this is to separate your analyte so that there's less chemical noise in the sample entering the spectrometer. This will reduce your matrix effects, improve ionization efficiency, and lead to better quantification. And it can also reduce isobaric interferences. So isobaric interferences occur when another analyte, but not your analyte of interest, has similar weight and charge to your analyte of interest. This can commonly occur with steroids, which are very similar molecules. So both gas and liquid chromatography will be discussed in detail in the upcoming future talk. Now I'll move on to ionization methods. So as I have mentioned previously, molecules are electrically neutral and cannot be manipulated using electric or magnetic fields. Therefore, this next step in mass spectrometry allows the conversion of molecules into ions. In the past, most ions were positively charged in mass spectrometry, but modern instruments can analyze both positive and negative ions. The process of ionization adds energy to the analyte molecules, and the method of ionization determines the amount of energy added to the analyte and the types of molecular species produced. So now I will discuss the ionization methods in a bit more detail. So electron ionization was the first widely used technique. It is still very commonly used in combination with gas chromatography. In electron ionization, analytes in the gas phase are bombarded with electrons obtained from a heated filament obtained in a vacuum chamber. And you can see on the slide, there is a diagram sort of summarizing that reaction. The energy imparted to the analytes is in considerable excess over that required to ionize organic species. So this is why this is termed hard ionization. And I have mentioned that term on the previous slides. The bombardment removes electron from the sample, so the initial product is a positively charged molecular ion. The excess energy is distributed among the bonds of the ions formed and often leads to the dissociation of one or more bonds and the ejection of radical or neutral species. So the resulting fragment ions will have lower masses than the molecular ion. And as mentioned previously, the fragmentation might be so extensive that no molecular ion is produced. This can make interpreting the mass spectrum difficult, as molecular ion is a very useful information about a compound. But the fragmentation is generally predictable and still provides useful information on the structure of the analyte. There are now multiple libraries of spectra that exist and can be used as a reference to help identify unknown analytes based on the predictable fragmentation. The next ionization method is electrospray ionization. So this method effect of placing a high voltage on a conductive solvent in which the analyte is dissolved and spraying the solution through a hollow needle. Ions of the charge opposite to that of the applied voltage are stripped from the solution. The charged nebulized particles are then evaporated continuously, first at atmospheric pressure, and then as they pass into a series of progressively evacuated chambers that form the connection between the source and the analyzer. The charge density of the droplets increases as they evaporate. And eventually a point is reached where the integrity of the droplets can no longer be maintained. The ions with the same polarity as the applied voltage are released into the vapor state. Electrospray ionization is compatible with polar compounds. It is also compatible with reverse phase liquid chromatography, as it is liquid-based and requires a conductive agent. And chromatography will be discussed in more detail in the upcoming talks. A very important feature of electrospray ionization is that as the molecular mass of an analyte increases, the ionization process often leads to the formation of multiply charged ions. The consequence of adding multiple protons to the analytes is that an envelope of charged species is formed, with each member separated by one charge from the next one. The generation of multiply charged ions has facilitated the use of mass spectrometers for the analysis of proteins by lowering the mass to charge ratio of large molecules. The next ionization method is the matrix-assisted laser desorption / ionization, also known as MALDI. And again, I have included little diagram in which, in this case, the MALDI system is connected to a time of flight tube as well. And I will discuss a little bit about this combination in the upcoming slides as well. But in MALDI, the analyte is first co-crystallized with an excess of matrix, of the sample matrix, that has a constituent aromatic component which is able to absorb photons from a UV laser beam. So when the dried analyte matrix mixture is exposed inside the vacuum chamber to a sudden input of energy from a laser pulse, the matrix evaporates, carrying with it the analyte molecules. The matrix forms reagent ions that then protonate the analytes. So this is the basic concept behind it. The next ionization method is chemical ionization. So chemical ionization is an example of soft ionization method, which has limited fragmentation and less energy is delivered. In chemical ionization, the electrons from a heated filament react with a reagent gas to form ions that interact with analyte, onto which a proton is transferred, producing protonated molecules. This type of ion is more stable as less energy is transferred than, for example, in electron ionization. Atmospheric pressure chemical ionization, known as APCI, is a type of chemical ionization method which is conducted at atmospheric pressure. And now I will briefly mention atmospheric pressure ionization. So atmospheric pressure ionization allows polar compounds to be analyzed. So ionization occurs at atmospheric pressure outside the vacuum chamber, and the resulting ions are transferred directly into the mass analyzer. Electrospray ionization is the most successful of the atmospheric pressure ionization methods because of the range of molecular masses that can be applied from small molecules to proteins. So other atmospheric pressure ionization methods include atmospheric pressure chemical ionization, which I've just mentioned on the previous slide, and also atmospheric pressure photoionization, which I won't discuss in further detail today as we don't really use it in clinical biochemistry labs. In atmospheric pressure chemical ionization, which is pictured on the slide, which has the diagram pictured on the slide here, both the analyte and the liquid chromatography solvent are evaporated, and the vast excess of solvent vapor is used to produce reagent ions by using a corona discharge to form an ionizing plasma. The discharge is released from the pin that replaces the filament used in electron ionization that would burn out immediately at atmospheric pressures. Ionization occurs through proton transfer from the reagent ions to the analyte, forming protonated molecules. Now, the next part of the talk will focus on the actual mass analyzers. So the role of the mass analyzers, as I have mentioned earlier on in the talk, is to separate ions according to their mass to charge ratio values and to focus and transfer these ions onto a detector or into a collision cell. The choice of the analyzer is critical as it affects the data generated, including the mass resolution, mass measurements, accuracy, and dynamic range that you will get for your mass spectra. There are several types of analyzers which you might have come across already. So those include quadruple or linear analyzer. You can also have ion trap, and both of those can be quadruple or linear, time of flight, magnetic sector, orbitrap, and ion cyclotron resonance cell. So both orbitrap and ion cyclotron resonance cell analyzers are known as something called Fourier transform instruments. So mass spectrometers separate ions in time or in space. In the quadruple ion trap, linear ion trap, and magnetic sector analyzers, operational parameters must be changed progressively over time, which is known as scanning mode, in order to obtain spectra. Those scanning instruments are in time systems because ions are collected successively over time. However, in time of flight and the Fourier transform instruments, those are in-space analyzers, which are non-scanning, and all the ions are detected simultaneously or almost simultaneously. So almost any type source can be coupled with any type of analyzer, except in MALDI, which cannot be coupled with quadruples because of their limited mass range. So now a little bit about the quadruple mass analyzers. So in quadruple analyzers, ions are separated according to their mass to charge ratio in an electric field generated into space within a set of 4 rods arranged in a square. And you can see that on the diagram. So it acts as a mass to charge selector. The opposite pairs of rods are connected electrically. Combinations of voltages placed on the rods generate fields that enable transmission of ions from one end of the analyzer to the other, with specific voltages being required for the transmission of ions for each mass to charge ratio value. The quadruple's ability to change its selectivity rapidly in milliseconds is due to the fast response time of the electric field oscillations. So ions that do not have the correct mass to charge ratio to maintain a stable trajectory are lost outside the quadruple's field. Now, something called triple quadruple has 2 analytical quadruples separated by a transmission quadruple. And I will discuss those instruments on the upcoming slides. It has several scanning modes that enable multiple tandem mass spectrometry approaches to obtain structural information about the analyte. So now moving on to the time of flight mass analyzer. So they are simple systems that measure the time it takes for an ion of a given mass to charge ratio to travel from the source to the detector. Packets of ions are pulsed into analyzer using an accelerated voltage where they travel through an evacuated tube to a detector. The ions are separated according to their momenta and therefore their mass to charge ratios. So lighter ions would travel faster than heavier ions. The next type of mass analyzer is the ion trap. So an ion trap mass spectrometer is an analytical instrument used to identify and quantify molecules by trapping ions and measuring their mass to charge ratios. It uses electric or magnetic fields to confine ions in a specific region of space, allowing precise manipulation, analysis, and detection of these ions. The ion trap confines ions using a combination of static and oscillating electric fields. Ions are ejected from the trap in a controlled manner, either by increasing the amplitude of the oscillating field or by resonant excitation. The ions are separated based on their mass to charge ratios and then detected. So now moving on to Fourier transform mass analyzers. So Fourier transform analyzers have the highest available resolution. So there are two types, as I have mentioned, known as Orbitraps or ion cyclotron resonance systems. In these analyzers, ions rotate or oscillate within a cell and are detected by recording of the electrical current that the passage of ions induces in the surfaces of the cell. The resolution attainable is inversely proportional to both the mass of the analyte ion and the time required to acquire the data. And this is just an example of an Orbitrap mass spectrometer. Now I'm going to move on to detectors. So once ions have been separated, they must be detected with the resulting signals amplified and stored. In electron multipliers, ions arriving at the point of collection hit a surface from which electrons are ejected. These electrons are then accelerated against a second surface from which an increased number of electrons are released. So this process is repeated, providing a cascade of electrons, resulting in a current, which is then amplified and recorded as data. So in Fourier transform instruments, ions move past a surface, and in doing so, induce an alternating electrical current, the frequency of which is proportional to the mass to charge ratio of the ions, while the intensity of the signal reflects the concentration of the analyte. The resulting image current has to be analyzed mathematically using Fourier transform methodology to actually visualize a mass spectrum. And now I will discuss in more detail tandem mass spectrometry, which I have mentioned previously when I was discussing the quadruple analyzers. So essentially, analyzers can be combined into tandem instruments, which can provide more information than single analyzer systems, which I have just discussed on the previous slides. So tandem analyzers are known as mass spectrometer / mass spectrometer instruments. And you might have already come across this terminology during your training. So you can utilize like analyzers. So for example, a triple quadruple systems, or you can utilize hybrid instruments. And an example of that would be a linear ion trap combined with an Orbitrapped instrument. So tandem mass spectrometry is based on analyzing the products of controlled collisions between selected ions and neutral gas molecules in pressurized collision cells that are placed in specific regions of the instrument so that the collisions occur between two mass analyzers. And just a little bit more on this on the current slide. So this is specifically a triple quadruple analyzer, and I won't discuss all four of the sort of scanning modes, but I will discuss the first one and the fourth one. So the triple quadruple has two outer quadruples, Q1 and Q3, which can be scanned. The middle quadruple, Q2, has no analytical function, but is a collision cell for the ions emerging from Q1. So radio frequency field is used to transmit ions to Q3 without any separation. In collision cell, the ions undergo collision cell dissociation through the interaction with neutral atoms of a reagent gas. You can have several possible combinations of scanning and statting modes for data collection that enable different analytical strategies. And this is what you can see on the current slide. So in product scanning mode, which is number one, Q1 is static, so fixed, and Q3 is scanned. So your ions of interest are selected in Q1, followed by transfer into a collision cell, where they undergo collision cell dissociation through interaction with neutral atoms of a target gas. The product ions, which are formed through fragmentation, are analyzed to their mass to charge ratio in Q3, producing a mass spectrum of the ions derived from the selected precursor ion. This is the most common tandem mass spectrometry strategy for obtaining structural information of unknown compounds. Then the other scanning strategy that I will mention is selection reaction monitoring. So in selection reaction monitoring, Q1 and Q3 are both static, which is also known as fixed. Q1 is set to pass only mass to charge ratio that corresponds to the analyte. So ions emerging from Q1 undergo collision cell dissociation in Q2, and the specific product ion is monitored using Q3, and that is also fixed, so pre-selected. So selection reaction monitoring provides highly specific and sensitive quantification of target analytes. And using this specific mode, the signal to noise ratio is improved, even although some ions are lost during the collision-induced dissociation in Q2. So the improvement is due to the low probability that the interfering species from your chemical noise and any co-eluting compounds which might not have been sort of fully addressed before your analytes enters the mass spectrometer during sample introduction. Those co-eluting compounds seen in the output from a single quadruple will have the same collision-induced dissociation ion as that selected for the analyte. So they will not be detected in Q3. And now moving on to some of the uses of mass spectrometry. So as I'm sure you are all aware, mass spectrometry has a wide variety of uses. It is important to know that compounds are analyzed in batch analysis rather than in the same routine way as in immunoassays, for example, in the clinical biochemistry laboratory. It can be used for qualitative analysis of known compounds to confirm their identity, or as I have mentioned, unknown compounds to determine their identity. It can also quantify known compounds or estimate unknown compounds of potential importance. So the choice of ionization method will have a dramatic effect on the spectra obtained. And the choice of ionization method might be influenced by molecular mass and polarity of the compound, and whether gas chromatography or liquid chromatography is used for initial separation. So some samples are clearly more complex than others. For example, in blood plasma, the compound of interest needs to be recovered from the matrix using solvent or solid phase extraction. And those extracts are still usually complex enough to need to be separated chromatographically prior to entry into mass spectrometer. The polarity of the compounds will determine whether gas or liquid chromatography should be used, and that will be discussed in the upcoming sessions on chromatography. So there are multiple uses for measuring analytes in the clinical biochemistry laboratory. I have given some examples on the slides, and I'm sure there are also many others. So now this is one of the final slides, a little bit about the internal standard. So an internal standard in mass spectrometry is a known compound added in a constant amount to a sample, blank, and standard solutions to improve the accuracy and precision for quantitative analysis. It serves as a reference for normalizing variation that might occur during the analytical process, such as sample preparation or injection, ionization, or detection. So an isotope labeled internal standard is preferred in mass spectrometry because it closely mimics the behavior of the target analyte throughout the entire analytical process. And this ensures accurate and precise quantification. And the peak height or area of the internal standard provides a reference against which the analyte signal is normalized. So the concentration of the analyte is determined using the ratio of the analyte's peak height or area to that of the internal standard. A calibration curve is then constructed using standards with known concentrations of the analyte and a constant amount of the internal standard. The response ratio, which you can see on the slide here, is plotted against the known analyte concentrations. The resulting curve is used to determine the concentration of the analyte in unknown samples by applying the response ratio obtained from the sample. So just a few take-home messages. So mass spectrometry is quite a complex topic, and it can be quite overwhelming when you start learning about it. I think the main things to remember as you start is that mass spectrometers analyze ions and that they measure mass to charge ratio. They can be used for qualitative and quantitative analysis. It's important to appreciate that there are different ionization methods, and they are and the one that you use is influenced by molecular mass and polarity of your analyte of interest. They can be coupled with gas or liquid chromatography, and mass spectrometry is obviously a very important and evolving field. So I'm very happy to answer any questions. If you could maybe type them in the chat, or you can always email me after the session as well. Here is my email address. And the final slide is just the images and references that I have used. So thank you very much for listening.